So I will talk about my project that is funded by Cure Cancer, and it's about looking at a novel inhibitor called venetoclax and why chronic lymphocytic leukemia fails this therapy. So a little bit more about my background. So I did my PhD in the Netherlands, in Amsterdam, already investigating leukemia. And after I finished my PhD, I was recruited to the Walter and Eliza Hall Institute in Melbourne to further investigate blood cancers. Luckily, I was supported by the International Leukemia and Lymphoma Society Fellowship. And this year, I was really grateful to get the Cure Cancer funding to further investigate this therapy. So my research is about chronic lymphocytic leukemia, so short CLL, and it's the most common adult blood cancer in the Western countries, and till this day remains an incurable disease. So we all have the immune system to fight off pathogens like bacteria and viruses that enter our bodies, and the main players of this are the lymphocytes. And you can divide the lymphocytes into two groups, the T cells and the B cells. But in CLL patients, all the B cells are cancer cells. And we normally, in a healthy human setting, we have a lot of B cells in our body. And these CLL patients have millions of cancer cells throughout the blood. So what does a cell makes a cancer cell? So normally in fast dividing cells, you get a stress signal to tell the cell to die because you don't want to have too much of these cells in the body. And this happens very controlled. In this CLL cell, even though it gets a stress signal, this cell won't die. And then these cells accumulate in the blood, the bone marrow, and the lymphoid tissues. And you can see this patient that has very bulky disease in the lymph nodes. So these lymph nodes are full of CLL cells. Why these cancer cells don't die is because they overexpress a pro-survival protein called BCL2. So these CLL cells have a lot of BCL2. And research she had done at the Walter and Eliza Hall Institute led to the development of a novel drug called venetoclax. And venetoclax really specifically targets BCL2. And if you treat these cancer cells with venetoclax, it rapidly kills these cancer cells. And venetoclax treatment is really a clinical success story in CLL patients. So the first patient treated in the world was here in Melbourne at the Royal Melbourne Hospital. And it was already 8 years ago. And if you just look at separate patients, just depicted over here, 3 patients, and then if you take the blood samples, you can see that the leukemic burden was going down. So after just a couple of months on venetoclax treatment, you can't no longer detect the CLL cells in the blood. And this is also what you see in this patient. So this patient had really bulky disease before it started venetoclax treatment. And just after a couple of months of taking venetoclax treatment, you can see that this bulky disease was completely gone. But venetoclax is not the cure for CLL. So even though patients do really well still after years of venetoclax treatment, in around 75% of these patients, the disease is coming back. So we call this resistance. And we really don't know what's happening. So why do these cells escape killing by venetoclax? And this is what my research is about. So what I do is I take peripheral blood samples before they start treatment and after they relapsed, and just look at what's different. So what's different in these cells that they escape killing by venetoclax? And that's what I do here. So normally what people do is looking at with novel, like old techniques. And actually what you can compare this with is looking at a smoothie. Like you maybe know what kind of flavors are in there. You maybe taste banana, maybe berries, but you don't really know what kind of berries. For this project, what I'm doing is applying novel technique called single cell sequencing. And instead of looking at a smoothie, I look at it like more as a fruit salad. So you really look at the individual cell and what's going on. And already, I'm not going to show like really complicated results, but what we already see is just within a patient, we see different mechanisms of resistance. So difference in different cells, a reason why they don't stop responding to venetoclax treatment. And ultimately, my goal is to identify all the mechanisms of resistance that have significant implications for how venetoclax is best used. First, maybe in preventing the development of therapy resistance, and second, by developing strategies to tackle resistance. And ultimately, this project should significantly improve the outcomes for patients with CLL. And lastly, I would really like to thank the Cure Cancer Australia and the Cancer Foundation for supporting this project. Thank you.